So thank you very much for the introduction and thanks for inviting me. So so my name is Yuval. I'm a group leader in the Department of Chemical Engineering, but as you'll see from my talk, my research is probably somewhere between chemical engineering, bioengineering, chemistry. But we're a very multidisciplinary group of sort of 25 or so members. So we really have experts in many, many different areas of science of engineering. And the overall goal of our lab's research is really to create synthetic cells from molecular components. So we start off with non-living components, and we gradually want to build more and more complex systems. And eventually, the aim is to build a synthetic life form in the lab. In the next sort of 10, 15 years, that's the goal. And the reason we want to build synthetic cells, as you'll see later, is we believe they're very useful tools, both with which to understand biology but also in various applications, including biomedical applications. So my research sort of fits in this area of synthetic biology. Sometimes, more recently, it's been called engineering biology. And it's basically a way for us as scientists, as engineers, to approach biology from an engineering lens. So can we alter living systems? Can we create new living systems and get them to do useful things for us? And classically, the starting point is a cell, is a bacterial cell or a yeast cell or mammalian cells, and we use genetic engineering and metabolic engineering techniques to get that cell to do things it wouldn't normally do. And it's a really, really hot topic. You know, people are saying this is the century of synthetic biology, the century of biology, and people are starting to think about synthetic biology as really underpinning the next industrial revolution. And the idea is that engineered biological systems will really have a direct impact on all sorts of things, on energy, in health, in the environment, in sustainability, and so on. And the UK government is pouring in huge sums of money into synthetic biology. Only recently, well, not that recently, when Rishi Sunak was still Prime Minister, he basically earmarked synthetic biology as one of three big areas which the UK government are investing in. One is artificial intelligence. The other one is quantum technologies, and the third is synthetic biology. So it's really viewed as being super, super important for the UK. So what are some of the areas in which sort of synthetic biology is proposed and is currently actually already having an impact? So one is here, I said, sort of artificial leaf technology. So people are engineering synthetic leaves, synthetic bacteria, synthetic algae, which can harvest sunlight and turn it into fuel, almost as a carbon capture sort of technology. People are using these synthetic engineered systems as new types of materials. So people are developing new types of fabrics that when you rip it, it self-heals and self-repair. And there are actually fabrics and shoes and belts on the market which are completely synthetic in origin, made of engineered living materials. People are looking at it as a manufacturing platform. So people are engineering bacteria to produce new types of commodity chemicals and drugs in a way which is very difficult to do completely synthetically, and fine chemicals too. For therapeutics, a huge, huge area for engineered biological systems is in therapeutics. So you may have heard of a new technology called CAR T cell therapy, where people are taking cells from the human body, re-engineering them outside of the human body, and then re-injecting them back into the patient and getting those cells to target cancer cells in a very effective and targeted way, using genetic engineering, essentially. Biomedicine and therapeutics for drug delivery and for vaccines. We're all aware of the COVID vaccines. These are mRNA vaccines. These are vaccines which contain engineered biological molecules, engineered mRNA, which are basically underpinning really the next generation of vaccines and anti-cancer agents. So synthetic biology really underpins all these applications and more. It's a sort of an inconvenient truth, let's say, or a dirty little secret when it comes to engineering biology. Is it actually possible to engineer biology, right? We all, well, many of you are engineers by training, um, but actually it's very difficult to look at a cell and look at biology from an engineer's perspective. So in this image here, this is a typical metabolic network of a cell, a metabolic pathway. So each node, there are thousands of nodes here. Each node is, let's say, a chemical species, and it's linked to another chemical species by some sort of reaction that happens inside a cell. And as you can tell, it's probably the most complex thing you can imagine. There are thousands of different chemical species, each interacting with one another to form an almost unimaginably large network space. So if you now have this system and you want to re-engineer it, if you now want to tweak, let's say, the bottom left-hand side of this metabolic pathway of the cell, you basically have no way of predicting how the top right-hand corner will respond. The cell will shuttle materials down different pathways, and it will sort of push back on the engineering procedures that you implement on it. So cells are living, cells are evolving, cells always want to do things that they want to do. They will always push back on what you, as an engineer, impose on it. And it should be intuitive to us that biology isn't like non-biology. You know, if you've seen the movie Jurassic Park, you know this Jeff Goldblum quote, life will find a way. These scientists in this movie Jurassic Park, no matter what they tried, life always found a way to do what life wants to do. It's very hard to control, it's very hard, and if not impossible, to predict biology, which in turn means it's very hard, if not impossible, to engineer biology in any traditional sense. So this is one of the problems. So how do we circumvent this? So one of the ways that we circumvent it is, instead of starting off with a really, really complex cell containing hundreds of thousands of distinct molecules, we want to build synthetic cells. So we start off with individual molecules of life, with lipids, with DNA, with enzymes. We make these molecules in the lab, or we purchase them from a chemical company. And then from the bottom up, we basically assemble a synthetic version of a cell that mimics some of the features and some of the behaviors of living biological cells. And we basically engineer it on the molecular level to reproduce the features and the behaviors that we associate with life. So when I talk about synthetic cells, this is what I mean. In our lab, a synthetic cell is some sort of capsule. It's some sort of spherical capsule. Mostly we make them out of lipid membranes because this is the building blocks of cell membranes. And inside these capsules, we can put antibodies on their surface to allow them to recognize the environment. We can put membrane proteins into the membrane to allow feedstocks to flow in, reaction products to flow out. We can build synthetic organelles within them, and these organelles can exist in different chemical environments. So you can start to perform chemistry inside. Different reaction steps happen in different compartments. We can put DNA inside our cells. These are called DNA genetic circuits or gene constructs. And just like you can program DNA of bacterial cells, we can program the DNA of our synthetic cells, and we can produce proteins inside our cells from the DNA template. And we can engineer communication between our synthetic cells and neighboring biological cells. So this is what a synthetic cell is. What are the features of it? What are the behaviors that we want to engineer into synthetic cells? So we can now in the lab engineer synthetic cells that can grow and divide. We can engineer synthetic cells that can take up energy from sunlight and convert it into chemical energy. We can engineer synthetic cells that can make decisions and sense and respond to the environment, and the response could be the production of a protein. We can engineer synthetic cells that communicate with the environment and move up and swim up a concentration gradient. So we're almost like one by one taking the behaviors that we associate with life, and we want to incorporate that and engineer that in a synthetic system. So as I said before, we want to do this because we want to use synthetic cells as a model to understand biology, but also in applications, okay? So one type of project that we have in the group is it relates to us using synthetic cells, which we construct from molecular components, as a way to answer biological questions. So typically a colleague from biology or life science will come to us and say, look, we're really interested in understanding this very specific biological pathway, but cells are far too complex for us to things are far too complex in cells for us to get to the bottom of how this pathway works. Can we use your synthetic cells as a tool to which we use to investigate our theory? So we build a synthetic cell, we take the proteins and the molecular components associated with that pathway, and we build a synthetic cell only with that pathway inside, away from the complexity of biology and the biological mess that's inside of a cell. And we can very systematically vary all components of that cell, the concentration of proteins, the composition of the membrane, the presence of salts and different buffers and different crowding agents and so on. And we can very easily do systematic experiments in a way that we can't do with biology. Because we can build cells from the bottom up and we have control over each and every feature of our cells, these are very useful biological models. The second sort of application, reason why we want to do this as engineering tools, as micro machines, essentially. For example, as sensors, which can alert us to the presence or absence of toxins in the environment, as delivery vehicles to deliver drugs and to deliver therapeutics to the target site, and as reactors to actually produce therapeutics and produce new molecules inside a capsule, followed by release. Okay. So again, not to labor the point, but another sort of reason for why we want to build synthetic cells really came to me when reading this book by Peter Thiel called 0 to 1. It's a fantastic book. So Peter Thiel is a pretty controversial character, but he's one of these brilliant multi-billionaire type guys from Silicon Valley. He's basically Elon Musk sort of best friends and on that sort of level in terms of wealth. And he's built several multi-billion dollar companies, including PayPal. And every year he gives a series of lectures to Harvard Business School about how to found a startup and how to build the next multi-billion dollar company. And it's called 0 to 1. And it's a fantastic book. He published the series of lectures in a book. It's called, which I encourage you to read. But half of that book, he basically says, whatever you do as a potential entrepreneur, my tips to you is never ever to invest in biology and never to invent a biotech company. Stay away from biology like your life depends on it. And this, as a biologist and as a biotechnologist, really offended me. But he has a point. And the reason he says this is as follows. So he basically says, we all sort of are familiar with Moore's law. Moore's law basically refers to this exponentially increasing ability of us as a human society to do computation. So every 2 years, we're getting better and every year we're getting better and better at producing new types of information technology hardware. Computers are getting better and better and faster. And this is an exponential trend going back to the 70s. And this is really what underpins our modern economy. And this is called Moore's law. But he says with biology and biotechnology, it doesn't follow Moore's law. Traditional biotech follows Eroom's law. So if you read Moore's law backwards, it's Eroom's. Instead of an exponential increasing trajectory, it's an exponentially decreasing trajectory. It's getting harder and harder and more expensive per unit time to develop a biotechnological product. And for those of us in the sector, we know, let's say for R&D, if you want to bring a drug to market, it's every 9 years, the cost of bringing a drug to market in terms of R&D costs is doubling. And this is not slowing down. And the reason he says for this is because you're working with living systems. So he has here a table where he compares and contrasts a biotech startup to a software startup. So with biology, you work with uncontrollable organisms, which are completely poorly understood. They're far too complex for us to ever understand properly. With software, everything's perfectly determined because you write software from scratch. You write code, line by line of code, and you know exactly what goes into the software. And by extension, everything is perfectly well understood. I hope you can see where I'm going with this. Synthetic cells and synthetic biology is a way to bridge this divide. It's perfectly determinate because you're making the organism from scratch. You know exactly what goes into it. Everything's super well understood because you're defining everything about that organism from first principles. Everything is definite, and you could apply engineering principles to biology in a way that you can't with normal living cells. The other big thing, which I think is driving our field forward, is this regulation aspect. So say you engineer bacteria which can sense arsenic in wastewater. If you want to take that engineered bacteria and drop it into a pond, the regulation will make it impossible. Currently, with genetic engineering, for various reasons, people do not want engineered bacterial strains or engineered living systems being thrown around into the environment. Why? Because cells can grow, cells can mutate, cells can do things that you don't really want them, that you don't sort of want them to do. But with synthetic cells, they're not living, they're not growing, they're not dividing, they're not mutating, and they're certainly not evolving. So from a regulatory point of view, you can still make the most of engineered biological systems without the drawback and without the associated cost. Same goes for therapeutics. For hundreds of years now, people have actually been engineering, academics that is, engineering bacteria which can have therapeutic effect. So imagine injecting a bacteria, that bacteria can move and swim towards a cancer site and target and kill the tumor. But the reason that this is not currently a product in the market is because bacteria can infect, they can grow, they can divide, and can mutate, and they're simply too toxic for this sort of applications. Many companies are trying to work on this, but still, there is no bacterial therapeutic on the market. But with synthetic cells, they can't grow, they can't divide, they can't infect. So again, from a regulatory and safety perspective, there are huge, huge advantages. Okay, so how do we make them? What are the sort of behaviors that we can engineer into them, and how you can start to think about using them in a therapeutic context? These are sort of three things I'll very briefly sort of mention in the remainder of my talk. So how do we make them? Okay, so the way we make them as engineers, we want to make them in a way that each and every one of them is exactly the right size and the same size and has the exact same content. We want really uniform cells because for any application, you need to do quality control on them and you need to make sure that they're uniform so that they have the desired effect. And the way we do this is using microfluidic technology. So we have a device. So I'm sure you're all aware of electronic chips, microelectronic chips. Imagine that exact same chip, which we make using the exact same technologies, by the way. But instead of sort of electronic wires, you have fluidic wires of the same dimensions, but instead of passing currents, they pass fluid flows. So we just go into the clean room, we design a device where we basically process different types of fluids. In this case, it's oleic acid, it's oils, it's water, it's buffers of different compositions, and almost like blowing through something that makes bubbles, we basically make bubbles where these bubbles are our synthetic cells. So this here is the output of our device, and you can see these are the cells that we make. We can make them continuously. We can make thousands of them a second, and they're all the same size. You'll see in this video here. This is me zooming out on the microscope. They're all the same size. They're all the same fluorescence. They have a uniform encapsulation, and that's basically how we make them in a very controlled way. The other thing about microfluidics is it allows us to control not only the size, but the internal architecture. So just like our cells in the body have organelles, you have a nucleus, you have mitochondria, plants have chloroplasts, they have all sorts of internal subcompartments that are very precisely defined. We can use microfluidics to build the exact same geometries. So in this case here, you can see two nuclei-like compartments in a cell. In this case here, you can see two cells which are linked together by a tube, a nanotube. And in biology, this is exactly what we get. Cells linked together by tunneling lipid nanotubes, they're called. In this example here, you see two or three membranes closely aligned with one another. In biology, we have the exact same thing. So you may know, let's say, a bacteria or plant have a cell wall, and outside the cell wall, inside, directly below the cell wall, you have a cell plasma membrane of a different composition. We can build the exact same thing into our synthetic cells. And what allows us to build these complex architectures are different microfluidic devices. So we've designed a whole sort of pipeline for making and manufacturing synthetic cells through different microfluidic modules. So in this module here, as an example, is a production module. In this case, here's a purification module. So here you see we purify the cells from all the junk that's a byproduct of the manufacturing process. So here, all the cells flow through this compartment, and using fluid dynamic flows, all the waste goes down here. So we can basically use this to purify our synthetic cells. We can trap them on our device, and then we can start to do systematic experiments on these traps. And we could do things like analyze them through things like flow cytometry. So this is a very useful as an experimental tool in an R&D and academic setting. It's all modular, so we can basically plug and play and mix and match these modules together. And this is something that we're now putting up on a website, which means that anyone in the world can copy our devices and can build their own microfluidic assembly line for synthetic cells, so to speak. This is another microfluidic device. In this case, we make these compartments within compartments. So we can load different materials into these different compartments. We can have two processes in these two compartments, and these compartments can communicate with each other and communicate with the environment through proteins and so on. So this is the assembly line. So the first part of the assembly line is here. So you can see we're forming these droplets, one white droplet and one black droplet, and these have different compositions within them, chemical compositions. They then flow down this meander, the snaking channel here, and around this is where the membrane assembles. The lipid membrane assembles at the interface of these droplets, and then we encapsulate them into in a second junction, which is what's shown here. And this is the output of the assembly line. You can see we're forming these things one by one, these cells. Each of them has two compartments within them, one black, one white. And this is the basis of our multi-compartment synthetic cells. Okay, so that's how we make them. But what can we engineer into them? So we're very interested in engineering behaviors into our synthetic cells and using some of the architectural complexity that we can build with microfluidics and translate that into functional complexity. So one of the things that we want to do is to design synthetic cells which can sense and respond to the environment. Think about a potential therapeutic, a synthetic cell that we engineer to sense a disease state. So it's a dormant cell. It senses the presence, let's say, of a tumor. It there turns on. It turns on by producing a protein of interest. That protein can be an anti-cancer protein. It then gets released from the cell. That protein can have its effect, can have its anti-cancer effect, and then that cell can then sense, for example, that threat, that tumor or that cancer has been eliminated, and it can then shut back down and recirculate. So these are some of the systems that we're trying to think about and build. So one of the things we wanted to engineer very early on is a cell that can respond to temperatures. Why temperatures? Because it's the most obvious place to start, and actually various disease states have elevated temperatures. So if this can be a way to target the therapy. So again, we turn back to biology. That's how we engineer these systems. So not to get too technical, but here we engineer the DNA. The DNA then produces RNA. And this RNA, because we can control the sequence of this RNA, we can basically control at what temperature it's either in the folded state or in the unfolded state. So we design systems so that at room temperature is folded. And when it's folded, it can't do its job. When the RNA is folded, the ribosome, which produces protein from the RNA, can't bind. So this whole thing is off. At a certain temperature, this thing unwinds and the ribosome can bind and you can produce the protein. So this is a way to use temperature as a trigger to produce a protein. Again, the beauty of biological engineering is that we can precisely control the base sequence, and we can precisely control the thermodynamics and the energy associated with binding and unbinding, which means that we can tune the temperature at which this thing unbinds and unwinds, and which you get the production of a protein. So what does this look like? So these are completely synthetic cells. There's no living system intact in sight. But we literally take all the components needed to go from DNA to RNA to protein, we put it into the membrane, and here you can see we can produce a green fluorescent protein. So these are non-living, but we put in the ribosomes, the polymerases, the amino acids, the nucleotides. It turns out there are 40 or so components, chemical components needed to do what all biology does. And we take those 40 chemical components, we put it into our synthetic cells, and they produce a green fluorescent protein, which is what these fluorescent dots are. And you can see at a low temperature, this whole system's off. You get no fluorescence. Where the arrows are, that's where the cell is, but it's not bright, it's not fluorescent. You raise the temperature, you get fluorescence. So we can turn this thing on, and this is how we analyze it. I'm not going to talk you through these graphs, but this shows us that we're getting what we think we're getting, which is a cell that can turn on and off depending on the temperature. So not only we can produce things inside, we can also release them. So instead of producing the green fluorescent protein, if instead what we produce is this membrane protein, it gives us a way of releasing material. So this is a membrane protein. So it's a protein that we produce inside. It pops into the membrane, and it basically forms a hole in the membrane. It's a membrane pore. It basically pops into the membrane, it forms a hole, and it lets things out in a size-selective way. So not only can we now produce the cargo, we can actually controllably release the cargo. And we use a technique called flow cytometry to measure this release. And you can also, on the microscope, see it. So here are many synthetic cells containing green fluorescent cargo, and you can see some of them start popping. this one goes, and that one goes, and that one goes over time, and eventually they all go if you leave it long enough. And I'll show you again. And this is basically shows that we can produce a membrane protein and we can release cargo stored inside. Okay, so that's the temperature as a stimulus, but we have many other stimuli which we have published and we are currently ongoing projects. So one of them, again, I'm not going to go into the details, but instead of temperature as a stimulus, you use magnetic fields. Why is that important? As we know, magnetic fields can penetrate tissue. So if you go into an MRI machine, there's a magnetic field there, and the magnetic field can very easily penetrate deep into the insides of the organs, which is why it's useful for imaging. But can we use magnetic fields where instead of for imaging, you very locally and directly activate the cells? So only in the disease state do you activate the cell. And this is again, activate the therapeutic. And this is a way to have a very targeted therapy with minimal side effects and more efficacious therapy. So that's magnetic field activation. We're also working on ultrasound activation. So just like you have an ultrasound for imaging, you use an ultrasound and have synthetic cells that can turn on in response to ultrasounds. Also hypoxia, oxygen levels, pH, and biomarkers. So we all know, so those of us who are medics may know that solid tumors are hypoxic. They have very low oxygen environments. Because they're so metabolically active, they use up the oxygen very, very quickly, and they're not supplied with enough fresh oxygen. So can we use that as a marker to tell us this is where the disease is? So can we engineer synthetic cells that can respond to hypoxia, turn on only when it encounters the solid tumor, and again, this being a very important targeted therapy. So these are all things that we're currently doing and we have done and we've published. If you want to read our papers, you're welcome to in the lab. Different types of stimuli. The final sort of example of a behavior that we engineer in the lab is motility. So movement. Movement up a concentration gradient, sometimes called chemotaxis, up a concentration gradient. So can we engineer a synthetic cell that can sense a high concentration of a molecule that's, for example, secreted by a tumor, and can it actively swim and migrate towards the tumor and actively accumulate there and target the tumor? Can we build a synthetic cell version of that? So here's a video to show a synthetic cell system that we designed in the lab that can do that. So here you can see a bunch of synthetic cells, and you can see when I press the video, they will all move simultaneously and in concert from the bottom to the top along the gradient of molecule that we apply. In this case, it's a polymer. Cool. So you can see here, they all move fairly quickly, actually, from bottom to top. And this is a form of chemotaxis, of directed movement up a concentration gradient. So these are the synthetic cells. They go some sort of shape change, which you see. They start off spherical, they go like a hemispherical shape, like a moon, and then they sort of shoot up like a comet, leaving a comet tail. So this is an example of motility and chemotaxis in synthetic cells. So again, I'm not going to bore you with the details, but there's all sorts of very fancy and intricate physical chemistry and flow chemistry which underpins this behavior, all to do with how the membrane rearranges itself around the droplet, which then causes a surface tension difference, which causes the motion. So it's something called the Marangoni effect, which some of you may be familiar with, and that's what causes this motion. So in this example here is motility, and then it's the final example of some sort of behavior that we engineer into synthetic cells is energy generation. So in our synthetic cells, once you form them, they're active and they can produce a protein, but very quickly they run out of energy. So you basically load them with all sorts of energy carrying molecules, but over time, the energy just gets depleted. So they have a lifetime of maybe 2-3 hours. If you want to extend it, the lifetime, you need to find another way of getting the energy in. The best way of doing it is using sunlight. So how do you convert sunlight into chemical energy? Well, how cells do it is using chloroplasts. So what we said is, okay, instead of re-engineering chloroplasts, why not just take a chloroplast directly from a living cell and put it into our synthetic cells? So what we do is we go to Sainsbury's or we go to a farmer's market, we buy spinach leaves, and we basically extract chloroplasts from spinach leaves, and we can put them into our synthetic cells. And the chloroplasts are still active. So when you shine light on the chloroplasts, they produce oxygen, And they also produce a molecule called ATP, which is the energy carrying molecule of life. And this ATP can be used to power chemical processes in the cell. So this is a population of synthetic cells. You can see they're all green, which means they're all active. And you see when it's dark, there's no activity. But as soon as there's light, you put it into sunlight, you get activity. And this shows that our whole system is active. And in this way, we can supply energy continuously into our synthetic cells. So the final example is something that we really wanted to do because synthetic cells have a lot of potential and promise, but as you can tell at the moment, it's super, super blue skies research. Okay, and what that means is, although we're funded by industry, everyone's fully aware that the actual applications for these synthetic cells are probably in the sort of 10 to 20-year horizon. But we began talking with pharmaceutical companies, so GSK and AstraZeneca, and for different applications. But they basically say that can we build synthetic cells on a nanoscale, on a much smaller scale, a much smaller regime, and this, for whatever reason, has much better and more immediate prospects as therapeutics. So you may be aware of the mRNA vaccines, which really the first mRNA vaccine was over COVID, the Moderna and the Pfizer vaccines. And these are basically mRNA molecules which are encapsulated in a particle called a lipid nanoparticle. And this is what it looks like. It's a simple spherical compartment with mRNA in them. And the mRNA gets delivered to the cell processes the mRNA, produces a protein which trains the immune system. And that's how the vaccine works. So this is a normal lipid nanoparticle, it's a spherical compartment. And you saw from the previous slides, we're very good as a lab at using various technologies, including microfluidic technologies, to form compartments within. Mhm.
Yes.
Okay. I don't know if that was aimed to me, but I will finish up. If the comment is, could you finish up? I will finish up in 2 minutes.
Yeah, no, I think that was somebody that managed to unmute themselves. So
Okay,
yeah.
okay, good. So in any case,
Bill, the speaker is muted currently. Can you help?
sorry, you can hear me?
Yeah, perfectly.
Yeah. Okay.
Yuval, I'm afraid you're muted at the moment on my machine at least. I'm not sure how to remedy that.
I can
So
hear
you have to unmute yourself.
As far as I'm aware, I am unmuted. I can see on my screen.
Can you unmute? Oh, Derek, you can hear the speaker, can you? I can't.
Yeah. So
what have I done
I think
wrong?
I think Alan's got his sound off. So I'd suggest you carry on.
Okay. So basically, what we want to do here in this project is to form our synthetic cells, but on a much, much smaller length scale. So normally our synthetic cells are 5 microns across. Oh, hello.
We can
Okay,
hear
we can
you.
hear you.
Sorry, but my thing is frozen.
Oh, okay.
Let me try one more time.
We can see and hear you.
I, for one, can't hear Yuval. Can anyone else?
We can hear.
We can hear.
Okay, so let me carry on. Now that's working. So we can now form synthetic cells on a nanoscale. So instead of a micron scale, it's a nanoscale. So just to give you some context, a microscale is, let's say, the width of a human hair. Those are normally the dimensions of our cells. But we've developed a new type of microfluidic technologies to make them on a nanoscale. So they're 1,000 times smaller than that. And what that means is, all of a sudden now, we can make them much smaller, and we can very precisely control the size. And what that means is, by controlling the size, we can control how well they fuse with cell membranes. So here we have 2 populations of compartments. One's small, 130 nanometers, and the other one's larger, 300 nanometers. And depending on the size, this affects the curvature of the membrane, and this affects the ability for that capsule to fuse with synthetic cell membranes and deliver their cargo. You can see when the particles are large, 310 nanometers, the cells are black, which means they have no fluorescence in them. And if we want to deliver a fluorescent agent, this is unsuccessful. But when we make them small enough, they all become fluorescent. So with microfluidics, we can control the size and we can control the performance of our drug delivery vehicles. We then play around with all sorts of chemistry, where all of a sudden now, again, we can form a compartment within a compartment. It's almost like, you know what, like a Russian doll. We have a doll within a doll within a doll. We can build lipid nanoparticle versions of that. A particle within the particle within a particle on a nanoscale. And we can control what's in each compartment. So what that means is think of a drug delivery vehicle, a particle which contains the drug. We want to design a system where we can release drug 1 at time point 1, at time point 2, release drug 2. And that's shown here. We're releasing drug 1 and then we're releasing drug 2 at different time points and at different temperatures. Why is that important? So I guess, again, if you think about vaccine applications or chemotherapies, you go back multiple times through multiple rounds of treatment or multiple applications of the jab. But many people do not come back the second time. There are also some compliance issues associated with that, also with antibiotics. Can you design a system where you just give one shot and at time point 1 it releases the therapeutic 1 and at time point 2 therapeutic 2? Our systems can do that. And the second thing they can do is we can release different drugs. So at time point 1 release drug 1, at time point 2 release drug 2. Again, for vaccine applications, very important. Why? At time point 1 you can release what's called an adjuvant, which is something that stimulates the immune response. It makes the immune system hyperactive, saying, oh, threat's coming. And at time point 2, you release antigen, the thing that the immune system is trained against. So again, for various therapeutic applications, we can now design systems which have multiple compartments, which can release different cargoes at different time points. And in this case here, we can actually produce something inside. So a particle on the nanometer scale where we can controllably mix chemicals together and then release them. So instead of performing chemistry in a vial, in a test tube, or in a reactor, we can perform it in a tiny compartment, potentially within the body, perform chemistry, and then release the product of that synthetic chemistry. So this concept is called synthesis at the site of action. So we want to synthesize the molecule at the target site, followed by release. Okay, so at that point, I sort of run out of time. I have 3 sort of take-home messages. The first is the technologies that we use in the group, both microfluidic technologies and membrane technologies, are really powerful tools to make new synthetic cells. That membranes and compartmentalization, along with genetics, can be used to engineer different types of behavior, be it sense and response behavior, motility, growth, division, and so on, which I haven't presented. And using similar concepts, we can now engineer analogous systems on the nanoscale, so much smaller size regimes. Okay, so with that, I'd just like to, oh, this slide is completely off. I'd just like to thank my research group, who have been really fantastic, and they've basically done all the work that I presented. And I'd like to thank my collaborators, Oscar Says, John Seddon, Francesca Cironi, other collaborators who aren't mentioned here are James Hindley, Claudio Cantini, and many more. So and thank you also all of you guys for listening. Okay, so yeah, thank you very much. I'm very happy to answer any questions at this point.
Okay, appreciate that. Thank you, Yuval. I just want to check if Alan is still with us. I think Alan was having some technical problems at his end. So,
Bill, hi, can you hear me?
Yeah, we can. Yeah, are you with us?
I can't hear you.
Okay. Right. I don't think you can hear anybody.
Can you take a...
Yep. That's fine. Right, okay, thanks for that. So, yeah, really fascinating talk, and I appreciate the level of detail and the breadth that you've covered in in about 35 minutes. So that's tremendous. We've got a couple of questions in the chat, but I wanted to maybe kick off with a question myself, because you mentioned the process that you go through for the manufacture, and you touched on purification, and I was curious to know, how do you detect and discriminate waste and take that out?
Yeah, so in our case, when we talk about waste, it's stuff that's not encapsulated, right? So what that means is that we have our cell, which is basically the membrane, and we have all sorts of junk outside the membrane. And the way, so what that means is that we can very easily purify based on size. So the stuff that we want is huge. It's maybe, well, on the scale of a molecule, it's huge. It's maybe 1 to 5 microns. And that's the flows that experience that sort of big particle experience is very different to the proteins, the DNA, to the lipids, to the aggregates that aren't encapsulated inside. So it's basically done on size,
Okay.
and that's how we discriminate waste from the thing that we want, which are the cells that we make.
Okay, that's perfect. Thank you very much for that. Olivia, you've got your hand up, so if you're able to ask your question, then that would be great.
I'm not a medic, so it's probably a very silly question, but you mentioned you were making it sort of on the web for anyone to use. Is it possible to make it used for evil purposes?
So for the field as a whole, it's a very, very important question, and it's something that we are all the time thinking of. So, if you're doing genetic engineering, yes. So like, people have, you could, for example, start to re-engineer viruses. So let's say viruses that have been sort of extinct and eliminated from the world, smallpox, polio, these sort of things. You could very easily design a DNA to produce these sort of viruses. There are all sorts of checks and balances and measures in place to stop you from doing that. But yes. But what I should say is our approach, which is bottom-up synthetic biology, we don't really do much genetic engineering. We sort of make the membrane and the capsule, and from the bottom up, we try to synthesize something that looks like a cell, that behaves like a cell. So that sort of stuff, luckily, is far too early for us ever to, it's not even being used for good applications concretely, let alone being misused. So it's stuff that it's a question that we need to ask, and we are asking, but it's the sort of thing that will only be important in the next sort of 10 to 15 years as the technology reaches maturity. But yes, this is something we always get asked, and we always have to justify, and we have to put measures in place to to sort of to address these concerns. But luckily, so one of the selling points of our synthetic cells is they're not living, they're not growing, they're not evolving, they're not mutating. So in a way, they're sort of easier to handle and to deal from that sort of aspect than a live cell biotechnology. There's less scope for things to go wrong, but they can still go wrong.
Thank you.
Can I just add to that? Is that on the boundary of what an ethics committee at the college would look at? Are you kind of within their remit or just outside it?
No, we're outside it, so they wouldn't. We're outside it, but if you come back in 10 years, when people have started to engineer these things to be autonomous, then yes, I'm sure this will fall under their sort of remit. But at the moment, it's not something that college or the research councils, we don't have to do much by way of ethics committees, unless we collaborate with people who do re-engineer bacteria cells and so on, and then we do that. Yeah.
Okay.
Do synthetic cells degrade? Or and so do they have a certain shelf life, and can the shelf life be changed, altered through engineering, so that
So yeah, it's something that we're looking into. So do they degrade? So physically speaking, in terms of their physical morphology, they're actually remarkably stable. Well, it depends what your benchmark is, but we find them quite stable. So what it means is that if you make our cells, these capsules that you see, and you can keep them in the fridge or the freezer, and in a month's time, they're sort of still there. So physically, they don't degrade. Mechanically, they don't degrade. Chemically, they would. So, DNA and RNA are quite unstable in water. So what that means is that over a period of a day or two, they will chemically degrade and be inactive. So how do we fix that? So one of the things that we're looking into is basically freeze drying them. So we take our synthetic cells, we put them through a lyophilizer, we turn them into a powder. They can then be stored in the fridge or in the freezer or at room temperature. And then after a month or a year, you rehydrate them. And that's basically the thing that we're doing to extend their shelf life, because at the moment, that is a big reason why, one of the reasons why this technology is still confined to academic labs rather than being used in the field. So yes, it's a great question, and yeah.
Because I was just thinking about what you were saying, where you have compartmentalized cells within cells, or layers of cells, and you have different active components in them, and you're trying to release them at different points in time. Those times could be months if you're delivering doses of a vaccine or something. So they need to last for months, perhaps even longer. You know?
Correct. If you want to use them in an in vivo environment as a therapeutic and have that release over a period of months, then degradation is one thing, but the other important thing is, what's the term? Yeah, your immune system has various ways to get rid of anything that's non-natural in the body. So people are, and we also on a chemical level, we engineer our cells essentially to make it immune and invisible to the immune response, so that the clearance time is much longer. It's still over a period of a week, it'll still get cleared, but it's not over a period of hours or days, which is without these chemical modifications, these things basically get cleared out and sent to the liver and extracted and degraded. But that's it's a big challenge which chemists are working to solve, and we're taking what they learn and putting them into our synthetic cells to prevent that.
Okay, thank you.
Yeah.
Right, thanks, Andre. There's a couple of questions in the chat. Is that David or Davide? If you want to ask your question, please come off mute, otherwise I'll ask it. So could you please elaborate a bit on the energy storage potential of the technology? You mentioned the sort of the biobatteries. How much energy could we store in these cells per unit of weight or volume? Do you have a comparison of energy density compared with batteries, petroleum?
Yeah, so we're very, very far removed from the energy density that you would achieve through inorganic means. So, we all think of photosynthesis as this amazing technology. Trees can take energy from sunlight, convert it into biomass, and so on. But it's incredibly, incredibly inefficient. Even the way cells do it and the way plants do it, the efficiency of carbon capture in those sort of systems is like ridiculously poor. And that's the way cells and biology do it. And we're trying to build synthetic versions of that, which is 100 times worse at the moment to what cells do, which is 100 times worse, which non-cellular systems do. So, in my view, in terms of applications for synthetic cells, at least, it's, because they're quite, at the moment, very expensive to produce, and B, they're not very good at things like, making things at scale and scaling up, though they're sort of more suited to producing fine chemicals or therapeutics where cost and scale aren't so much of an issue compared to the energy sector. So, yeah.
You kind of touched on cost there. Could you give us an idea? Because I remember when we visited the Molecular Science Research Laboratory, I remember Oscar asked us a question: how much would all the chemicals in your body cost? And he talked about the amazing cost of lipids. So the constituent parts, your precursors.
Yeah. So it's interesting. Like, I think to make a population of cells at a density which you need, for example, to have a therapeutic effect, at the moment it would cost somewhere like $200 per dose. Which, when I first started thinking about, I thought it was quite expensive. But then when you think about the state of the art in cancer therapy, which is a CAR T cell therapy, that costs I couldn't believe when I looked it up. It costs £500,000 to buy, maybe £300,000 to make. And that's like, if you need it, that's how much health systems around the world are willing to pay for efficacious treatment. So for things like therapeutics, they're competitive. They're sort of similar in cost. So orders of magnitude similar in cost to like a mRNA vaccine, which in terms of the building blocks, they're made of lipids and they're made of RNA. So that's the sort of cost. So at the moment, I think it's maybe $20 a dose. For the vaccines. Yeah.
Okay, that's good, even though it's in the research area.
Yeah.
Okay, we got a couple more questions. So Asma, happy for you to come off mute, or I'll ask the question. Do you have any examples where you successfully use synthetic cells to kill or target tumors? I guess you're not in the kind of clinical area just yet, but have you done
We're
any
not
of
in the
that?
clinical, but we have sort of in vitro data. So when we culture cells on a flask and we show they have sort of yeah, we do sort of live dead assay and cell death assays, and on that sort of level, you know, they do what they're meant to do and what we engineer them to do. So that's normally the sort of experiments that we run. In terms of animal models, again, for ethical reasons, it's not, you know, the technology is not there to justify us starting to stick it into sort of mice and chimpanzees and stuff. It's sort of still too early for that, for us to justify that. So, but sort of cell culture level, in vitro cell biology stuff is stuff that we sort of work on at the moment.
Okay.
Are the main are the main applications at the moment more in the medical sphere, or do any members of your research crew are looking into perhaps using them to build biological logic gates or something, and then go more towards the use of like computers?
In my research area, it's mostly biomedical applications, but you know, other groups who are in related areas who are working a bit more on the DNA side of things are working on it on as an information storage medium. You know, if you think about like, you know, the cells in your body, think about the information that they store, a single cell and a single DNA in the cell. It contains information that was originally created back in the origins of life, right? You still have information and signatures that have persisted for billions of years. So as an information storage medium, cells are amazing. They store huge amounts of data, and and they reproduce and pass on that data with high fidelity over multiple generations for long term. So if you think about a hard drive, if you wanted to get a hard drive to survive for thousands or tens of thousands or hundreds of thousands of years, no chance. If you wanted to engineer bacteria to do that, actually maybe you could engineer life form, which the purpose of the engineered system would be simply to store materials over multiple decades and millennia. So yeah, but that's not my sort of research area. But for synthetic biology as a whole, there are big companies which are looking at storing data inside cells as a storage medium. Yeah.
Thank you.
Okay, we've two questions left, and then we'll wrap up. And I'll choose the last question in the list, and then I'll come back to Peter's question, which will be a good one to wrap up on. So, do you use mitochondria for energy production?
We don't. We mostly you can. We mostly stick to chloroplasts because we're interested in getting energy from the environment, so from light. Mitochondria process energy in a slightly different way. Other people have, I believe. The other thing about but we sort of don't. The other thing about mitochondria is quite hard to get a hold of a large quantity of good quality mitochondria. So you normally have to get liver samples, and it becomes a bit fiddly to work with to extract mitochondria. Yeah, but we haven't really played around with mitochondria too much.
Okay, right, thank you. Peter, if you want to come off mute and ask your question, because it would be a really good one to wrap up on, or I'll ask it on your behalf.
No, I'm unmuted, I think.
You are unmuted. Yeah, you go ahead then, please.
So in practical terms, this sounds a fascinating new technology from anything I've seen before. I had no idea you could do these sort of constructions. Have you got any imaginative targets for 2025 as we approach Christmas?
Yeah, it's difficult, you know. As in an academic lab, things move quite slowly. So in a year's time, it's I don't think anything much can change. But you know, who was it? Bill Gates said, you know, people always overestimate what they can do in 2 years, but underestimate what they can do in 10 years. So, you know, in 10 years' time, let's take that sort of horizon. You know, it's a very rapidly moving research area. So 5 years ago, there was no one working in this area. You know, there's Oscar Says, and there was me doing a PhD with him, but it was really like slim pickings, which was good for us. But now it's become such a hot topic. There are hundreds of groups worldwide now working in this area, and huge groups, Nobel Prize winners, who are now pivoting to this research area. So the pace of advancement is huge. So one of the big things that people want to do is synthetic cells that can grow and divide over multiple cycles, because that would really be a big landmark. That would basically mean that you made a synthetic life form in a way in the lab. And as I think it was Olivia said, at that point you start getting ethical considerations, safety considerations, and so on. But once you can get synthetic cells that can grow and divide, you can do things like directed evolution and these sort of things. So the big landmark for the area is a synthetic cell that can autonomously self-sustain without external input, can grow and divide over multiple generations. And I probably think we're probably not 10 years away from that, probably 15 years away from that. But let's see. And obviously AI is a big thing. People are now looking at incorporating AI technologies and AI platforms into synthetic biology. So it's a really big topic, which will really help speed the field along. Yeah.
Thank you very much for a fascinating topic.
Yeah.
So I'd like to wrap up because we're close to 5, and we try and promise that people can go off and do their evenings on a Friday. So so very much appreciate the talk this afternoon, Yuval. It's been fascinating. I think when we chatted earlier on, you kind of explained some of your background, and the kind of the move into the bioengineering area is clearly a fast-moving discipline. So very much appreciated. Just for our participants, we've recorded the event, so we will put it onto YouTube if anybody wants to replay it. And thank you to that. So just with that, I'd like to ask people, we try and do a virtual clap. If people can unmute themselves or raise hands on then YouTube, then we will like to thank Yuval for a great afternoon talk.
Thank you.
Thank you.
Okay. Thank you for that.
Thank you very much.
Thank you very much, everyone.
Thank you.
Right,
Thank
you.
Fascinating talk. Thank you.
Yeah,
Very
brilliant.
much.
Thank you. Thank
Okay.
you. Thank you. So